It's a pleasure to introduce Johannes Elferich from UMass Chan Medical School. We may all know him because of his plugins for system. So he's developing approaches and methods in cryo-EM image processing, and also interested in neurons and cancer. And today we're going to hear about high-resolution cryo-EM reconstructions from vitreous sections.
Here we are. Let's do it like this. So thanks, everybody. I want to thank the organizers for inviting me. And today I'm very excited to talk about a project where we kind of try to figure out whether an alternative way of getting thin slices of cells would work maybe better or equally well as FIB-milling, which is kind of the predominant technique that we use nowadays to make these fantastic images. And before I jump in, I want to briefly talk about why I was so excited about being part of that project. So one thing I like to think about is the brain, right? I mean, who isn't thinking about that? And so this is a movie made from this really cool data set that comes out of the Microns project, where they take I think it's a plastic embedded brain tissue, and they cut it in these long ribbons and high throughput image everything and put it together and train all the neuron paths. And you get this really rich data where you can annotate all these neurons, and all these yellow dots are synapses, right? Places where one neuron talks to another. And that's essentially how we think. And I don't think anybody here would fight me on this when I would say that in order to really understand it is we need to image each one of these yellow dots, right? And really understand how our molecules organized, where the receptors, where's neurotransmitter being released. And so what would be really great is to image these synapses with cryo-EM, right? And that is a very innovative idea, which is why in like the last 2 years, there have been like paper after paper of people doing cryo-ET of synapses, right? And as a representative example, I'm going to show some work that I was involved in. So this is work together with Eric Guo and Aya Matsui and Kathy Spangler in Portland, Oregon. And what you see is a lamella from vitrified mouse brain. And so in this case, a FIB and the ion beam was used to basically ablate the material on top and below of this thin slice. And so we can now start to zoom in. And sometimes you see these places where you have synapses. So what gives them apart are these little round things. So we now go through the tomograms. So the presynaptic side is like filled with these little vesicles full of neurotransmitter. And then here in green is shown the postsynaptic side, where there will be receptors that sense them and then create an electric signal based on that. And so in this study, what we did is we labeled some of these receptors called AMPA receptors with a little gold particle, so they stick out very nicely in the EM. And if we now zoom in on like this part of the postsynaptic membrane, we can, by looking at the positions of these gold particles, which we can define very accurately because it's like a high intensity signal, we can get an understanding how these receptors are organized. But if we really want to understand it, the problem is, right, it's cut off at the bottom and the top. And we don't know, are we looking at the edge of that synapse? Like what's really going on then? But that part was literally vaporized, right? It's sticking to the walls of your FIB-SEM. So the question is there another way of making these preparations where we don't lose all that like really valuable information? And the other way is CEMOVIS, which is also not a new idea. It goes back to like I think to the 70s or 80s almost. And what you do is basically, quite simply, you take a knife and shave off these thin slices of like a block of vitrified material. And then they form these, they kind of stick to each other, I guess. I've never done this, but they stick to each other and you get this ribbon, and you can put that ribbon on a grid and image it in a TEM. And this is how the grid looks like. So as I said, I have not generated this data. This was done in Bern, Switzerland, mostly by Marek Kaminek, Wanda Kukolski today learned grew the yeast that is on these cells, and then Benoit Zuber is the PI, and this all happened because Nico went there in person and forced them to do it because we really wanted to have these samples. So if we again zoom in a little bit, you can see that in this case the slices were laid over like a very standard holy carbon. I think it was coated with platinum, but that doesn't really matter. So now in all of these holes, you see they're all covered with a thin slice that we can actually see through an image. What you might already notice is that there are these lines that kind of go perpendicular to the ribbon. So they were parallel to the edge of the knife. We zoom in even further, we can start to make out the slices of yeast cells that are in that ribbon. And you can also now see these cutting artifacts that are in these samples. And then zooming even more, we can like take high resolution micrographs of these samples that are now at pixel sizes where we can really do the high resolution cryo-EM that we all want to do. And one thing you might notice, there is in this micrograph, there's a membrane, and kind of connected to these aberrations, you can see the membrane sometimes seems to be torn apart. And I think this is the main thing why people have not done this as much as we should do, because in FIB that doesn't happen, right? In FIB, the ultrastructure is perfectly preserved and it's all beautiful. But what we were wondering is what's with the high resolution information, right? How does that compare? Is that maybe also gone, or is it maybe well preserved and we could actually use these slices? So the first thing we do every time we get a micrograph, right? We look at the power spectra, and that looks actually fantastic, right? We could fit the ton rings up to very high resolution, 3.1 angstrom. You see that characteristic pattern of thick samples where sometimes the ton rings seem to disappear. And from that, we can estimate that the sample was about 175 nanometers thick, which is a good thickness for like a cellular EM sample. What is interesting is that the nominal thickness while they were being cut was 100 nanometers. So it seems like these samples end up thicker in the microscope than you wanted. But the problem, of course, with the power spectrum is it has high resolution information, but it might be high resolution information of junk, right? It doesn't tell you if you can actually do something with it. And to figure that out, we use the technique that we like in the lab that's called 2D template matching. And there's a very silly animation to kind of explain you what we're doing. So we have like our beautiful micrograph. So this is from FIB-milling, so the membranes look beautiful. And we take a known structure, like the 60th subunit of the ribosome, and we just calculate the cross correlation of every possible projection of that molecule with that micrograph. And if that cross correlation becomes significant, we know there was a ribosome at this position, and we also know the orientation, right? Because we know which orientation gave rise to the cross correlation. And so over time, we can build up like a 3D model of the ribosome inside of this sample without ever having to tilt the stage. And tomography is amazing, but I think we all know that it has its issues, right? It's very complicated to process, and there's probably some information that gets lost during the alignment. So this is kind of more a straightforward way of testing which information is actually in that micrograph. And this animation is still going on. So you might think that the problem with that is it's super computationally expensive. It takes forever to run, but our computers are getting faster. So it's pretty doable nowadays. And so we knew that this would be a great technique to assess the sample because of the brilliant work of Bronwyn Lucas, who previously used it to basically quantify the sort of damage that occurs during FIB-milling. So on the left image, what you can see, all the dots are detections of 60S subunits, and they're colored by their SNR, basically on their cross-correlation. And you can see that cross-correlation is worse at the bottom and top surface of the lamella, which we interpreted as the ribosomes there getting damaged by radiation damage. So this is super neat to figure out. And the other thing is an idea of using 2D TM results to calculate reconstructions. And normally everybody would shout at you when you do that, Einstein from noise. But what you can do is you can like strategically omit certain stuff from the template. So whatever you reconstruct in these locations is presumably a true signal. So we adapted this idea for this project. So the template we prepared for 2D template matching is of the 60S subunit of the yeast ribosome. And we omitted two parts of that template. For one, we omitted subunit L34A, which is kind of at the periphery. And we cut out like a box where we removed all the atoms within a central cube with a side length of about 40 angstroms. And you can see if we look at the slice to the density, it's just the ribosome has a huge hole in the middle. Nothing is there. And then our first question was, can we actually get cross correlations on our CEMOVIS data? And we were elated to say, yes, we got plenty of what we call detections, right? Statistically significant cross correlation peaks. So this is just an example of the image you saw before, and overlaid all the ribosomes we found. And this is like a view of the cross correlation map. So you can see there, here they're three teeny tiny dots. Each one is a high cross correlation. And if you look at a like 3D plot of some of these peaks, you can see these stand out really, really, really well from the surrounding noise. So we have very high confidence in this being true detections of ribosomes. And if you want, I can tell you later on over coffee a bit more about what the statistics are. But we are very, very, very sure that these are true detections. And so the first thing we did is, of course, compare this to FIB-mill data. So we used the data that Bronwyn Lucas produced in her paper, where she looked at radiation damage in FIB-mill sample, which was also of yeast, and first asked how many detections are there per unit area. And we plotted this as a function of sample thickness. So because you would think this also kind of depends on the volume you're looking at. And what was quite striking is that while the sample thickness is, I think, comparable between the two samples, is that in some of the FIB images, you just have way, way, way more detections than in the CEMOVIS images. So there are many ways to explain this, but probably the best guess is that there are maybe areas of the CEMOVIS sample that are getting damaged, and we have basically cannot detect ribosomes anymore. This is further supported when we look at the SNR scores. So we have two different measures. One is the Z-score, and the other one is called refined SNR. If you're interested, I can explain that to you, but the results are kind of the same. It shows like quite robustly the SNRs we get in the CEMOVIS sample are quite a bit worse than in the FIB-mill sample. And this is, it's not quite clear if that's really a fair comparison, because these data were taken on different continents. The pixel sizes are slightly different. The detector was different. And the FIB sample, there was cyclohexamide added. It's like a ribosome inhibitor, which shouldn't affect the 60S subunit, but it might. There were, of course, also different methods of vitrification, different operators of the microscope, lots and lots and lots of variables, right? But I think the effect is striking enough to think that this might just be the case, that on average, there is probably more sample damage in CEMOVIS than in FIB-milling. But the question remains is, can you do structural biology with that? So we went ahead and calculated the reconstruction. And on the top, you again see the template, with the point being the center is blank. There can be no template bias there. But in the reconstruction, which you can see in the bottom left, in red, I've called it the density in these areas. And there's very, very strong, clear density apparent there. And if you look through the size of the density, it's like the hole wasn't there. So we're very sure that's like a true reconstruction of the density of the 60S subunit in this area that we could obtain in CEMOVIS samples. Of course, the first question, everybody wants the number. What is the resolution? And everybody wants to see the FSC curve. So I went ahead and calculated the FSC curve only in that central area. Basically, I cut out the map in this area. And here it is. And if you look at the map versus map, it goes through the magic value at around 3.1 angstrom. And the map versus model is at around 3.2 angstrom, which sounds great, fantastic. But again, we can talk about it. It's an FSC curve, right? This is not gold standard. There was no gold standard procedure involved here. This is just randomly selecting half particle sets and calculating cross correlations. So they might have spurious cross correlations for all kinds of reasons. I think the true test is looking at the density, right? Where that makes sense. So this is the density that we obtained for L34A. So in the middle is a model. The structure is very well known, right? And on the left is like the segmented out density from that map. And I think it looks pretty nice. It's probably not 3.1 angstroms, but you can clearly follow the backbone. They're like bulges for side chains. And of course, I did the thing where I looked for the prettiest region of the map and kind of compares with the model. And you can see some density for arginine side chains. This map, I think I filtered through 3.5 angstrom resolution and applied a B factor of -70. That's kind of the only map modification I did because we want to give like as raw an impression of the resulting data as possible. So it is definitely noisy. And then this is the map of like the central cube. Again, we think it's a pure reconstruction of the density. And I think it looks pretty nice. There are many projects where it would have killed for a map like that. You see density for a lot of the bases very well. And I think you could really do biology with it. We should probably have tested if Model Angelo can build in this, because this seems to be like, at the current state of the field, kind of the threshold we want to cross, right? So this is definitely something I should do. But yeah, I think this is a nice map, and turns out you can definitely, I think, do structural biology with CEMOVIS. The one thing we wanted to figure out is, since we thought there's damage, right, where's that coming from, right? Is it also on the top and the bottom of the surface? So on the left, you see, again, I basically recreated Bronwyn's data using our template to make everything comparable. And you very nicely see the lower SNRs at the top and the bottom of the FIB-mill sample. But if we look at the side view from the CEMOVIS sample, there's, I think, no such pattern. It just seems to be randomly good and bad SNRs. So whatever the damage mechanism is, it probably somehow penetrates the sample. And then we spend a lot of time like just staring on like the plots of the detections on the micrographs to kind of figure out whether we can like see a pattern, like where in that image do we get detections or not. And so the first thing we did is we kind of bandpass filtered the images to kind of see these crevices very well. And then every one of us had like immediately an idea. It was like, oh, they're like strings in the dark areas. And somebody else like, oh, they form string in the light areas. And we were really confused. And then I just did the thing where I plotted a histogram of all the pixel intensities in the bandpass filtered images and overlaid the intensities like at the spot of detection. And they overlap perfectly. So whether the image is bright or dark locally does not seem to predict where we can get our detection or not. But the more we stared at them, I became convinced that I think I can see that the detections occur like in these strings that are parallel to these crevices. So the question was just, how do we quantify that? And so what I came up with is I first tried to find a way to like measure the axis of the crevices. So I do that by basically asking a projection along which direction will create the highest variability of image intensities. And then, so we basically can calculate it at every possible angle. And then where I get the highest variance, I call this psi crevices. So that's the angle of the crevices. I show you two images here from two different grids. So the knife was kind of cutting from a different direction. And you can see in one case that angle is 36 and the other one is -12. And then I did a clustering analysis that's called Ripley's K or sometimes Ripley's L function. And what you normally there is you just ask if you have like a point pattern, you basically draw a circle around every detection and ask how many other detections are in that circle. And with that, you can kind of figure out whether points are more spread out or more clustered than you would expect by chance. And so we modified it by using an ellipse instead of a circle. So the ellipse is basically an elongated circle. And so we basically asked if we count the detections with an ellipse and then rotate the ellipse around, at which angle do we get the most clustering? And when we plot that out, that agreed very well with the previous plot of the psi crevices. So when I say that the psi clustering equals the psi crevices. And this was consistent over like the top 100 images where we had the most detections. So what I think I can say is that in CEMOVIS samples, you can get 60S detections in elongated clusters that are parallel to the knife direction. So what does that really mean? So this is kind of the simplified model I have, and we can fight about whether this is what's actually happening, but this is like an example in a FIB-mill sample. So what we're looking at is the ion beam came like this and milled the sample, and we're looking like at a cross section. And what we know is there's damage kind of at the top and the bottom of the lamella, and in the middle, everything is super nice and well preserved. But in a CEMOVIS sample, what might be happening is as the knife cuts along here, the sample has to fracture because it needs to kind of bend away from the block in the ice. And these fractures, whether through a local melting or some other mechanism, are probably damaging molecules in their vicinity. So you get also damage around these fractures, which in effect means you get these patches that are elongated in a direction parallel to the knife, where presumably molecules are relatively undamaged, and we can do structural biology with them. So in summary, does CEMOVIS produce higher quality cell slices than FIB-milling? No, not yet, at least. Because the thing is, people have done a lot of FIB-milling, and the method by now is very optimized, whereas CEMOVIS probably was neglected somewhat. But is the quality high enough for in-situ structural biology? Yes, for sure it is. You can get very nice maps with it. And CEMOVIS does have advantages, right? The imageable area, right? In a FIB-mill sample, you measure in square microns, like tens, maybe hundreds of square microns. But the imageable area in like a CEMOVIS sample is almost square millimeters. It's a lot more. And then, of course, you have that continuity thing. You have consecutive sections of the sample that presumably in the future we can stack together and make real volumes out of it. So what I think we should do is we should use 2D template matching to kind of really, I think it's a great way to get quality scores for CEMOVIS samples, and we can use that to get better at doing CEMOVIS. Maybe the knife needs to be faster, it needs to be slower. We might different coatings for the knife, different coatings for the grids. These are all now things where we have a metric now to optimize that to get the best preservation of high resolution signal. And then what we should do is we should combine these CEMOVIS samples with montage cryo-EM acquisitions. So we basically image that whole ribbon on a grid, stack the slices up, and we can do cryo volume TEM at super high resolution. That's my dream for the future, at least. So with that, I want to thank a bunch of people, the Grigoriev lab, mostly Nico, who took care that that data was being acquired, our collaborators, Eric Wu, and then Wanda, Benoit, and Marek, who did the actual data thing. And I want to thank you for listening to me, and I'm happy to answer any questions.
Thank you, Johannes. Questions from the audience? As well, there is a question. Aaron, if you can please pass the microphone.
Cool talk, Johannes. You should be commended for trying to go against convention, so congrats. Have you tried matching something else apart from ribosomes, say like microtubules, actin, membranes?
Not in these samples. There is other data out there suggesting that you can do a high resolution structural biology in CEMOVIS samples. I think you should talk to René Frank about that. And I think there was also a study using 2D crystal scattering. So yeah, there's data on stuff other than ribosomes, but 2D template matching becomes quite hard on things that are smaller and less abundant about ribosomes, and that's definitely ongoing work that is challenging.
Yeah. A quick question on the subsequent section. What you have shown is for the first section, or you have really studied how the situation goes when you study if the deformation actually propagates and something else happens?
At the moment, we have done, unfortunately, none of these things. But yeah, that is definitely something that we need to figure out. We also don't know whether materials gets lost on the knife, right? Yeah, we should get on that.
One more question from Slack, from Julia Zanetti. If the goal is to image thicker regions, how do you correlate features on the successive slices? And somewhat related, could you think of a different grid support that would leave more support-free regions to be imaged and increase chances to have a complete Z stack?
Yes, I mean, the correlation thing is, so I imagine it would be very hard to correlate one specific region with another one. So I think what we should do this montage thing where you image the whole slice, and then it's much easier to image two whole slices together, right, than one specific feature, which might get lost because it's over a grid bar, or these are all problems. So yeah, the sample support is another great question, right? So we ideally want to have, right now there are these holes, right? And we can only see in these holes. So we want larger holes, less grid bars to really minimize the amount of sample that we can no longer image. We will probably never be perfect, but we can try to be as good as possible.
Yep.
Hey, you mentioned that the power of CEMOVIS is that you have a non-destructive technique, and sorry, I'm over here, and you get much thinner samples than you would with FIB-milling. Saying that the second point there is probably the most powerful of the two, considering you lose ribbons from the microtome to the TEM, and the carbon support probably obscures quite a lot of it. Is there a reason you went for 100 nanometers rather than thinner?
No, I mean, the 100 nanometers is, we need at a starting point, right? I don't think, this is precisely the thing we have to do now, right? We have to see how thin we can go. Does going thinner make things better? Does it make it worse? There's probably, like with FIB-milling, there's probably an optimal thickness. Because with FIB-milling, there's an optimal thickness because of that problem of radiation damage. If you go too thin, you start to damage most of your lamella. So the same thing will be true here. There's probably an optimum, and we now have the tools to figure out where it is.
Thank you.
Another question, and the last one.
Over here. Great talk, thank you. Do you think that you would see the same pattern of damage if you did STA? Do you think you would struggle to detect in the crevasses, or do you think that a 3D reconstruction would?
Yeah. The problem with STA is because you have so much more upstream processing, right? With aligning the tilt images, it will be hard to know whether you don't get good STAs because you couldn't align the tilts very well, or because you couldn't align the subtomograms. Yeah, I don't know how these crevices, I think I've heard mentioned before that they change under the electron beam. So if you do the higher doses you would need for tomogram imaging, that might also be a problem if they change during your tilt area. So I think the power of 2D template matching here is that it's such a more straightforward form of data analysis. But for a lot of projects, you might want to do tomography if you, for example, want a 3D model of your membranes, right? So this is definitely also somebody should start doing.
Thank you.
Thank you. Let's thank Johannes again.